Hello, this is Matt Dean with A+ College Ready, and today we're going to talk about cell signaling and cell communication. Cells must be able to communicate with adjacent cells, and in some cases with cells located in other parts of the body. We're going to first talk about the case where cells are communicating with local cells, or cells that are adjacent to each other. Oftentimes this is done through what are called intercellular junctions, junctions between two cells. There's a couple of examples you need to be familiar with. Plasmodesmata are communicating intercellular junctions in plant cells that allow them to transfer materials and signals between each other. And you've also got similar things in animal cells called gap junctions. These are really important in places like the cardiac muscle cells where they help the cells to beat in rhythm. Let's look at a couple of pictures and examples of these. So here are plasmodesmata. These green outlines are the cell walls around the plants. But these little yellow tunnel-like structures, these are the plasmodesmata. So they go through the cell walls and allow for the transmission of materials and signals between adjacent plant cells. Here's a very similar thing in animal cells. So we're looking at a cell membrane in here and a cell membrane in here. And these little purple structures are the gap junctions that are allowing these adjacent animal cells to communicate with each other. There's also a situation where membrane-bound cell surface proteins connect and help two adjacent cells communicate. This is really, really important in the immune process. We will have talked about this in the immune system video, which will be posted here soon. Here's an example, though. So this cell is a macrophage. The macrophage goes through and engulfs germs, just like this one. Maybe this is a virus. It will break it down through a process called intracellular digestion, using the enzymes from its lysosomes to do that. And then it's going to take some pieces, some of the antigens from that virus, and it's going to do what's called antigen presentation. It's going to display some antigens from that foreign invader on its own cell membrane. And we see that happening here. So this green structure is called an MHC2 protein. And it's used to present that antigen, which is what we're looking at there, to other parts of the immune system, in particular to helper T cells, in order to make them aware of the presence of the invader and to activate them to go out and attack that invader. Another example of local signaling is what we call synaptic signaling. This happens between adjacent neurons. And these neurons, they don't actually attach, There's a small gap between them called the synapse. And they transfer information back and forth, at least signals back and forth, through the use of these little chemical signals called neurotransmitters. So we can see that here. This is called the presynaptic neuron. And we have an electrical signal, an action potential going down that presynaptic neuron. When it gets near the end, there's an influx of calcium ions into that neuron. And that facilitates the release of the neurotransmitters into the synapse, which is what this little gap is here. The little dots represent the neurotransmitters. They cross the gap. They bind to these little green receptors on the postsynaptic neuron. When they do, that causes those receptors to open up. That allows for the influx of typically sodium ions into that next neuron, and that's the beginning of the electrical signal in the postsynaptic neuron. So those are all examples of local signaling. Now there's also cases where where cells may need to communicate with cells that are pretty far away. There's several ways that can be done. Oftentimes it's done with signals. Signals that may be released into the blood or the air. Most of these signaling molecules interact with receptors that are located on the surface of the cell. Those are called extracellular receptors. And the signals that work this way are called ligands. That's an important word that we'll see throughout this unit. Typically, they're made out of proteins, which means they're too large and charged to actually pass through the cell membrane. So they have to interact with receptors on the outside. Some signals, though, are made out of lipids. Steroids are an example of a type of cell signal composed of a lipid. Estrogen and testosterone are two important types of steroids. Because they have a lipid nature, they can cross through the plasma membrane. They can interact with the receptors that are on the inside of the cell, called intracellular receptors. And the receptor and the steroid together act as what are called gene regulatory proteins. They go through the cell, into the nucleus, stimulate specific genes, and they can cause a fairly slow but a sustained response. Some examples of long-distance signals are hormones. Hormones are signals that are released from an endocrine gland into the bloodstream. They travel through the bloodstream and bind to receptors on specific target cells and transmit a response or a message to them. We also have pheromones. Pheromones are substances released by an animal, maybe in the urine or in the sweat or other body fluids. They travel typically through the air, and they send signals usually related to either reproduction or attraction. These can travel many meters. We also have nervous system signals. Now here we're not talking about at the synapse. We're talking about the actual impulse that travels through the nerve. The longest nerves in your body can be up to a meter in length, so fairly long distances. Next, we're going to talk about the steps involved in the cell signaling process. And it's usually broken down into 3 steps. Step number 1 is called reception. And this is when the signal binds to the receptor. This often causes the receptor to change shape. And most of that change in shape happens just inside the membrane. It's called a conformational change. And this transfers the signal message from outside the cell into the internal part of the cell. Step 2 is very important. It's called transduction. In this case, the signal is converted to a form that can bring about a response. This part of the process is often a multi-step process. Sometimes it's called the signal transduction pathway or the phosphorylation cascade. And the reason for the complication and the many steps is that at most of the steps along the process, the signal is being amplified. So by amplification, we mean that the signal is being spread out throughout the entirety of the cell. The last step is the response. So the signal causes some type of response. Often it can include things like turning on or turning off a specific gene or set of genes, turning on or turning off specific chemical reactions, maybe stimulating the breakdown of some kind of substrate, or in the case of nerve cells, opening or closing a specific ion channel. So here we see a diagram of step 1, reception. So we've got our ligand. It's binding to an extracellular receptor located in the cell membrane. It's going to bind. That's going to facilitate the change of this receptor, the change of shape of this receptor, which is going to move the signal, not the actual ligand, but the signaling process to the inside of the membrane. I think we looked at this already. It's important to note that these ligands bind specifically to a receptor. They fit together sort of like a lock and a key, which makes receptors very specific to a particular ligand. Step 2 again is transduction. So once the signal is now inside the membrane, a set of proteins will then transfer the signal throughout the cell, causing it to be amplified, eventually bringing about the response that needs to happen. Remember, this is sometimes called the signal transduction pathway. As I said a minute ago, it's a multi-step process. The benefit of these multi-steps is that a small number of signals or ligands can activate a large number of molecules at the end of the pathway. That's what we mean by amplification. You could almost think about this like a crazy chain of dominoes, where if you tip the first domino, it's going to tip multiple dominoes. The chain is going to split in multiple ways. By the end of the pathway, you may have knocked over thousands and thousands of dominoes. It's amplification. Many of the steps in this process involve phosphorylation, which is the transfer of a phosphate group typically from ATP to a particular protein, causing that protein to be activated. This process often involves a whole category of enzymes that carries out these phosphorylations, and those enzymes are called protein kinases. So many of the steps in this process, many of these steps are carried out by protein kinases, enzymes that activate other proteins by transferring a phosphate group and therefore energy to them. Here we see a transduction pathway or a phosphorylation cascade. So notice that our signal molecule bound to the receptor, that helped to activate some type of relay molecule, which then activated a protein kinase. That protein kinase then phosphorylates a second protein kinase and activates it. That one then may activate a third one and a fourth one, until eventually some type of enzyme or process is activated at the end, which causes the desired response. Now this diagram is a little misleading because it makes it look like the process is linear, and that one of these phosphorylates one of these. What oftentimes happens at each of these steps, one of these may end up activating multiple protein kinase 2s, which means now instead of having one path, the path splits. And this is where the amplification happens. So by the end of this process, thousands and thousands of this active enzyme at the end may be activated, and that can cause a large, fast response. Again, step 3 is the response. Some type of cellular activity is activated. Happens at the end of the signal transduction pathway. Several kinds of responses that might happen in response to cell signaling. The opening or closing of an ion channel. The breakdown of a substance, like a glycogen, a polysaccharide. One of the effects of adrenaline acting as a cell signal is to cause the breakdown of glycogen and the release of glucose into your blood so that you have more energy for the fight or flight response. Responses may also activate enzymes. They may cause the synthesis of enzymes or other proteins. They may also affect gene regulation. They may cause the turning on or turning off of certain genes. Another thing involved in the process of cell signaling are these structures called chemicals called secondary messengers or second messengers. Their job is to transmit the message, the signal, from just inside the membrane throughout the rest of the cytoplasm. The secondary messenger that you're going to hear the most about is a chemical called cyclical AMP or CAMP, C-A-M-P. It is usually associated with a type of receptor called the G-linked protein receptors. It's built from ATP, and we're going to talk more about CAMP in a second. Some other chemicals that can act as secondary messengers in the body include calcium ions, especially involved in the nervous system, and a chemical similar to CAMP called CGMP. We'll get to CAMP more in a second. Let's talk about some receptors in the body. One of the most important kinds of receptors are called G-protein linked or G-protein coupled receptors. These are a type of extracellular receptor. They're the largest and most diverse group of receptors found in eukaryotic organisms. They can bind to ligands that help to send signals about odors, pheromonal kinds of receptors, even hormones, even neurotransmitters. So they are very important. Once a ligand binds to one of these G-protein receptors, a GTP molecule, which is much like an ATP, is attached to a part of that receptor called the alpha subunit. That subunit, the alpha subunit, then breaks free and goes on to activate another protein. That protein, that activated protein, then usually starts to create the secondary messengers like CAMP. This starts the signal transduction pathway and the process of amplifying the signal throughout the cell. One of the maybe disadvantages of G-protein linked receptors, each one of them can only activate one specific signal transduction pathway at a time. So here's a picture of a G-protein linked receptor. So we see here the ligand binding to it. This alpha subunit now gets GTP attached to it. It breaks away, slides down the surface of the membrane and activates the second enzyme. This enzyme, at least in a lot of pathways, is called adenylyl cyclase. Adenylyl cyclase. And this enzyme will take in ATP and convert that ATP to cAMP, the secondary messenger. Another type of receptor is called the receptor tyrosine kinase, or RTK. These receptors are also extracellular. They respond to signals like growth factors and other locally released proteins. They play an important role in the regulation of cell growth. That's probably the most important area. Cell differentiation and survival. Insulin receptors are an example of this type of receptor, an RTK. When signaling molecules bind to RTKs, they cause two neighboring RTKs to combine together to associate, forming cross-linked dimers. They connect. This activates the enzymatic activity, what's called the tyrosine kinase activity in the RTKs through phosphorylation. Each one of these RTKs can phosphorylate multiple pathways. This allows them to activate multiple signal transduction pathways at a time, and they can stimulate multiple cellular responses. So they can cause fast, large reactions, faster and larger than a G protein linked receptor might cause. So here we see a receptor tyrosine kinase. So notice we have these two neighboring receptors. When the ligands bind, they attach, they form that dimer. Each one of these locations at the bottom can phosphorylate different proteins, activating multiple signal transduction pathways at one time, causing fast, large responses. Another type of receptor is the ligand-gated ion channel. These are found mostly on excitable cells, like neurons, but they can also be found on muscle cells and receptor cells. They tend to convert chemical signals into electrical signals. The way they work is they maintain by active transport different concentrations of ions, at least the cells do, on each side of their membrane. And these concentration gradients can be used to generate an electrical signal. So what ends up happening with ligand-gated ion channels is that a neurotransmitter will bind to the receptor, the ligand-gated ion channel receptor. They'll cause it to go through a conformational change. That change typically causes the channel to open, and it allows the movement of some charged ion to go into or out of the cell, thus generating an electrical signal. Because remember, an electrical signal is simply the flow of some positive particle. So here we see a ligand-gated ion channel. Here's the channel. Here's the ligand. It binds into it, almost like a substrate will fit into the active side of an enzyme, causing it to change shape and open up. The ions flow through, and that's the beginning of an electrical signal in this cell. Maybe it's a nerve cell, so it's causing the beginning of an action potential. Here we see a little bit more about secondary messengers. So again, here's our G protein linked receptor. Our signal, our ligand, in this case epinephrine or adrenaline, binds in. It causes the alpha subunit, sometimes you're going to see it called the G protein, to get GTP associated with it. It slides down the membrane, phosphorylates and activates adenylyl cyclase. Adenylyl cyclase takes in ATPs and makes cyclical AMPs. And again, this diagram makes it look like they only take in one ATP and make one cAMP. But in actuality, lots and lots of these cAMPs are made by this one enzyme. So this is where the beginning of the process of amplification happens. Those cAMPs will activate protein kinases, which will start the signal transduction pathway, ultimately causing some type of response. Here's a diagram of what cAMP looks like. So here's ATP. Remember, that's adenosine triphosphate. This is adenine, the nitrogenous base. This is ribose, the sugar. And these are phosphate groups. Over here, there's three of them. That's why it's ATP, adenosine triphosphate. Adenylyl cyclase rearranges this a little bit and converts it into cyclical AMP. So notice 2 of those phosphates are removed. We get a little circle right in here. So it's called cyclical AMP. And this particular chemical, this nucleic acid, will now act as the body's most important second messenger or secondary messenger. And that's it for cell signaling for today. Hope this helps.